Hi guys, grade 12s, welcome. We're back with Life Sciences today, Life Sciences Live, and what we're doing is we're doing genetics and genetic engineering. Now genetics is quite complicated and we're going to make sure that you've got this so that you're not like past learners that have battled with genetics, because we're going to make sure that they get, without a doubt, you can get 100% for genetics. Don't let them tell you that it's difficult. We can make sure that you can do it. So stay tuned, and we're going to be doing some exciting stuff on genetic engineering as well.
I don't know if you watched, but I hope you've all got hearts as strong as an ox, okay? I'm going to quickly go wash my hands, and I'll see you in a minute.
Mindsetters, welcome back. Learn Extra Live, Life Sciences, grade 12. This is for you. It's all about genetics. We're going to finish off the ideas of Mendels Laws. We didn't quite finish it last week, and then we're going straight into some genetic engineering. So, Kathy, please take it away.
Hi, guys and girls. All right, well, we're nice and clean now. Hands all sorted. Ox heart. By the way, John tells me some of the learners want to know if it was a real heart. Oh my gosh. People, we are not allowed to dissect real life people parts. You know, the only time you're allowed to do it is if you're doing anatomy and you're at med school and they are bodies that have been donated to science, and even then you have to treat it with respect. I mean, can you imagine if we just chopped someone's heart out? Whoa. All right, but the ox heart, the human heart, is just much bigger. Remember, your heart, for the grade 11 learners that are still watching, is the size of your hand. Right, so that's the size of the heart. It's not really a big thing. Okay, little thing. All right, so here we go. Genetics. We've got Mendels Law of independent assortment and inheritance. For all intents purposes, the only reason this law is important is for you to understand the difference between the law of dominance and segregation. So what he said here was that each characteristic, every characteristic that you have, your skin texture, your nose, the shape of your body, whatever the case is, you pick something. Each characteristic that you have in your body is controlled by allele genes. In other words, one allele from your mother, okay, is from the mom, and one allele will be from the dad. Okay, those allele genes on a separate pair of chromosomes. Remember, one chromosome from mom, one from dad. Mendel, or male or female, Mendel stated that the different pairs of chromosomes segregate, means they separate, independently in a hybrid. So each characteristic or trait is sorted out independently. So what they're basically saying is, if your dad has brown hair and blue eyes, and your mom has brown hair and brown eyes, okay, the genes for eye color and the genes for her hair color, and the same for the chromosomes that have your dad's eye color and hair color, they're going to separate independently. So you could end up with brown hair and either blue eyes or brown eyes. But the fact that your hair color, eye color, etc., is not blonde, must be blue eyes. Brown hair must have brown eyes. Black hair must have very dark eyes. It's nothing like that. Each characteristic separates independently, and that is during meiosis 1, during metaphase, you've got random assortment. All right, and also remember, during prophase of meiosis 1, you have the crossing over. And that's when a piece from your mom's chromosome will break off and join a piece from the father's chromosome, or the male chromosome and the female chromosome. Okay, so you must just basically know his law, and it's the law of inheritance. So it's saying each characteristic, which is controlled by the allele genes on the separate chromosomes, they are going to segregate or separate independently when an offspring is created, and it's as simple as that. Okay, this is just for your interest. Meiosis took place, and here we're looking at two different characteristics. The characteristics that Mendel looked at was whether the R is for whether the seeds were rough or smooth. okay? And the Y was whether they were green or yellow. Right, so, and based on that, you've got your little chromosomes, they separate out, you have independent assortment, and they assort as they want to. So you can either have small r, small y, capital R, capital Y, small r, capital Y, and capital R, small y. So it's all the different combinations, and you are always going to have your offspring, when you calculate it, if there's one characteristic, it's 2 times 2, which is 4. When there are 2 characteristics, remember they've each got 2 gametes and 2 options or 2 crosses. So you end up with 4 outcomes or 4 possible outcomes. So what do you have? If one has 4, then 2 will have 16 different options. So you've got 1, 2, 3, 4, and 1, 2, 3, 4. Right, so you'll have 16 variations. It's either going to be a dominant green, and it's going to be smooth, and so you're going to go through all these various options. Independent assortment, all right? Now we go with sex-linked characteristics, and they love asking questions on this in an exam, okay? There are certain things that are sex-linked. Now remember, in males, we have XY chromosomes. And for the sex chromosomes, we have, of all our chromosomes in our body, remember we've got 23 pairs. 22 pairs are what we call autosomic or somatic chromosomes. They are for your body. And then 2 chromosomes, or 1 pair of chromosomes, is your sex chromosomes. And the sex chromosomes is what determines whether you are male or whether you're female. So I'm a female, so my 22 pairs of autosomes are there, and then I have 1 pair of chromosomes, and that pair of chromosomes will be XX. John is a male, right? And you can see he's a man. Very cute man, but he's a man. And John has an XY chromosome. Right, so the Y is actually an X with a little short leg, which means, and I just need a bit of space here, which means if we have, let's get a color, if we have XX, which is the female, and we have XY is the male. This is just a little tiny short leg over here, so that's why they call it a Y chromosome, all right? So there is nothing here to compensate on this female's structure X chromosome there. Here, there is, but there isn't. So anything, any gene characteristic that sits here on the female chromosome, there is no allele on the male chromosome, and that is what causes the problem. That is why these poor guys end up suffering from all kinds of things, which us girls carry. Okay, so remember that. And it's going to be, guys are generally colorblind. I mean, very few males that I know actually are good with colors. Matching clothes, worrying about whether their outfit suits what they're wearing and whether the belt matches the shoes, blah, blah, blah. Why? Guys don't see color. And very often you'll find your moms are buying your dad's clothes, and you will end up buying your boyfriend's clothes one day. Why? Because we say, oh, they don't have taste. Actually, they do have taste, but very often they end up with the color blindness gene. Why? Because it's carried by the female, and oh shame, there is no allele on the male chromosome here to compensate. So whether it's recessive or dominant makes no difference. It's going to come out in one out of 4 of the of the children of the offspring. 50% of the boys, or in fact in the total 25%. So how do we write this? Anything that has to do with being transmitted sexually or on the sex chromosomes, we are going to write the XX for the female, the XY for the male, and we're going to allocate a letter to whatever it is we are checking. In this case, it's hemophilia. Now, people, hemophilia is a lack of the ability to clot your blood. And Shane, it's the poor boys that end up with it, not the moms. And in fact, Alexander the Great suffered from this. He was the Tsar, in fact, he was the son of the Tsar of Russia. And Shane, his mother did everything she could. And there's even an old song called Rasputin, or all about Rasputin. And he was a really slimy thing. But he was also a doctor, and he did a bit of magic and all kinds of other nonsense that they have in the stories. And he was able to help that son and prevent him from dying. Okay, but because he was the heir to the Russian Empire. And anyway, it all fell flat anyway, so don't worry about that. But that is a very good example of hemophilia. So go onto the internet, even if you have to go into Wikipedia, although I'm not too hot with Wikipedia because a lot of the stuff there isn't edited. You can just edit whatever you want to in. But have a look there, and you'll see the whole history of hemophilia and how it affected the Russian Empire. And it's history. It's something you should know. It's something you can talk about at a party and sound intelligent. Okay, so here we go. Here's the work. H tells you it is normal. And it's normal, and it is dominant. This recessive should actually have gone here. So here, hemophilia is recessive. All right, it's just the slide that looks a bit funny. So H, capital H, it's dominant. And remember, we always write the dominant characteristic with a capital letter, unless they tell you to do differently in an exam. Okay, and hemophilia, the disease, okay, it's a genetic disease, is recessive, which tells you what? You need 2 little h's, or 2 recessive genes, for it to come through in a female. But in a male, hello, what do we have? He has no arm. There are no alleles to compensate. Okay, so the female carrier, X, capital H, she's normal, and X, small h. So they're telling you that she has the recessive gene, but this would be complete dominance. All right? And the hemophilia is recessive. Now, with the male, we have a normal male. So we have a normal female. He's carrying both genes in her allele set. We have a normal male, and he doesn't have hemophilia. He's normal. All right, so he's got the dominant gene on his X chromosome, and he is normal. So everyone's happy. And the 2 of them, P1, get together, and they're going to have a baby. So fertilization takes place, and we separate the chromosomes or the gametes out. So we're going to have an X with a capital H. We're going to have an X with a small h. That's the female. And we are going to have the male with his capital H. And we're going to have the little Y chromosome here without the little arm. And if it doesn't have the little arm, it can't have anything. So there's no compensating allele, but it's okay because it is the dominant gene. All right, everyone's happy. Your gametes, there are your gametes. The gametes are going to undergo meiosis. All right, so there are your marks there. Fertilization takes place. So what do we have? X capital H, there we have an absolutely normal dominant female. She is homozygous. Okay, homozygous, pure, absolutely perfect, dominant. And we have X capital H, X small h, and what do we end up with? We end up with a capital H and a small h here. Now, this little girl, because remember it's a female child, XX, she is going to be a carrier. Okay, she's not going to have it because remember it's complete dominance. So she's not going to have hemophilia, but She's carrying that recessive gene. Okay, now, in the male, hello, Y plus XY, there's my little boy, and that one's normal. But look at this. Shame. Y and the X plus the H, and this is going to be the affected child. So, in the 4 gametes that are produced, and the variations of reproduction that takes place when fertilization occurs, what have we got? We've got 1 in 4 chances that it's going to be a girl, and she's going to be absolutely normal, 2 dominant genes. We get for hemophilia. We're going to have a 25% chance, or 1 in 4, that it's going to be a little girl, but she's going to be carrying the recessive gene. And then we're going to have 50% of boys, 50% of girls. That's normal. But we say, hang on, little boy here, he's got the dominant gene. He's okay. But 25% of a chance for this child being affected, should be affected.
It's affected.
Affected, and there he has hemophilia. And shame, you know. I mean, the mom and dad don't even know it, and there's this little thing that's now a hemophiliac. And for those of you that have read, I think it's Bryce Courtney's April Fool, he had a son, and they only found out that he was a hemophiliac when he was a couple of years old, and he used to go for blood transfusions because they end up not being able to clot their blood. They don't produce enough red blood cells. It's a whole big mess. And you know, and they don't produce, it affects their red blood count, it affects everything, but they can't clot. So just a little bump, and they almost bleed to death internally. And he ended up with HIV because it was before the time that they tested all the blood. And they gave him a transfusion that gave him HIV, and that's what the book's about. So, you know what? Read, people. Reading's important. Okay,
can before we go into the next example, can we go to a break?
Just before we do, I just want to show them now with the F1 generation. I don't want to leave it because I need for them to see where their marks are. F1 generation, genotype, remember, is genetics. What do you see genetically? 1 in 4 are going to be that. 1 in 4, it's as we explained it. Your phenotype, 25% normal females, 25% normal males, 25% carrier female, but unaffected, and 25% male that has hemophilia. And with that, John, we can go to ad break.
Guys, that's really great. I hope you're following that. Sex-linked diseases and genetics on sex-linked, they are going to come up in your exam. So make sure you've got this spotted. I'm going to put up onto Facebook the question and the solution, so watch out for it there. We're going to a short break. We'll be back straight after that.
Okay, people, please, you will, as John said, without a doubt, you will get a sex-linked disease, sex-linked mutation in an exam. They love asking it, all right? We also have to go to pedigree charts at some stage too, which is very important, because if you get a pedigree chart, piece of cake to work out. Here we go. I've put these slides on. It shows you the combinations when you have an affected father with an X-linked dominant gene. So it's not on his Y gene, it's not on the Y, it's on the X chromosome. The female, unaffected. She's got 2 recessive alleles, she's happy. And what do we end up with? They show affected and unaffected. So here we have an unaffected son, we have an affected daughter, we have an unaffected son again, and an affected daughter. So why? Because it's on that XX chromosome. So when we get the X plus the X that makes the girls, they are going to automatically end up with that affliction, that disease, whatever it is, whatever that chromosome is carrying. All right, next one, and John's going to put this on for you as well. Here we have an affected father, but it's a recessive allele. So we had affected father with dominant, a affected father with a recessive. Remember, there's nothing on the Y. And then we have an unaffected mom. So what are we going to have here now? We have an unaffected son, XY, an unaffected daughter, XX, an affected son, and an affected daughter. All right. And our last version is if we have, now we've had an affected father with an unaffected mother and an affected mother. Now here we've got an unaffected father and an unaffected mother, but what is she doing? She is the carrier. So she's carrying this. All right, so dominant allele sits on the X chromosome. Okay, here we have X-linked and it's recessive. That's the one we're looking at. And Shan, look at our babies. So here we've got 3 characteristics now. We have affected, we have unaffected, and we have the carrier. We're going to have an unaffected son, an unaffected daughter, an affected son, and an unaffected carrier daughter. And this is a perfect example of hemophilia. Okay, this is how it's carried. So you end up with a process where you've got a little girl and she's going to be unaffected, but when she has babies, she's going to be sending and possibly causing her child to be to have the affliction if it's a boy. Okay, now, before we go to frame shift mutations, Taboch was here again, and he's so sweet. He's like our own special on-the-job cameraman. And we're looking at genetic variations. Now, how do genetic variations occur? And how can we do it as human beings? Easy. We interfere with God's creation all the time. Why? Because we want to find cures for diseases and illnesses. We want to make things suit us in this world. So we sort of speed things up and we have artificial selection. And when you get to the whole evolution process and we do diversity, you are going to have to be able to explain and link genetics to the fact that we have artificial selection and we have natural selection. Now, natural selection would be the genes do their thing, and slowly but surely over time and depending on the environment, new species are created. Okay, that's natural, that's normal. They change. I mean, we've got peppered moths that are white and peppered moths that are brown in the same forest, literally. But they've changed and they're the same species. And then we get clever and we start playing with polyploidy. Poly means many. Ploidy, we're now looking at our chromosome pairs. So instead of being diploid, means one from mom, one from dad, one from male, one from female, and we have our chromosome pair, we're now going to bring in a third chromosome so that there are 3 for a specific chromosome. We have that when we have Down syndrome, all right? And there are 3 chromosomes for chromosome 21 instead of the homologous pair. Okay, in animals, if there's any genetic engineering there, we can't fiddle or play with any of the chromosomes because as animals, and this goes for all animals, we can't deal with polyploidy, having more than one pair of chromosomes per set, okay? But in plants, we can. And this is where genetic engineering is amazing. Now, have a look here. Look at what I've got. See John again. I mean, he's such a star. What can I say? Okay, I'm going to show you this. Here we have grapes and grapes. All right. This grape, take a nice juicy one.
Sorry to interrupt you, Kathy. Can we just look at the label at some stage of that one? Yeah. Okay. Cool.
Are we allowed to?
Yes, because there's no advertising on that label.
Oh, okay. Perfect.
That's why I got that.
All right. We've got that. And now we're going to take a grape from the other bunch, which is on the plate, and we're going to slice it open, and there we go. So this is from the plate, and this is from our box. Okay, now this grape is what drives me insane and makes me want to give my son up for adoption. All right, why? Because he takes those little round things and instead of like any other human being, putting them in his hand, politely, of course, and putting them in an ashtray, he spits them out on my carpet, and he says he forgets. So that's my son, and this causes a lot of problems in our house. So I love the genetic engineers because they came up with that version. And what do we have here, people? In this one, we have got my son's favorite and my carpet's not so favorite, pips. Okay? So if I plant those, they're going to grow into little plants. And those little plants will be wonderful genetic variations of the mother and the father, or the mother plant with the male and the female parts that made it. Okay. In this structure or little fruit over here, there are no pips. There's a little thing here which is just sort of like a promise. It's just tissue. All right? So there are no pips, which means that in our second boxed fruit, all right, what do we have? We're sitting here now with fruit that doesn't have anything on the inside. So if I take this and I plant it, am I going to get offspring from the original plant? No, and there will be no genetic variation. Why? Because the scientists have taken the perfect combination of chromosomes that they want, all right, and they've put them together to produce this. Now, John wants us to go onto the label here, and Toboko, if you can do that while I try and find my scalpel that I dropped. Okay, just can you zoom in on that? I'm getting my glasses so I can actually see what's here, because this is written like in minute ant language. All right, it tells us here it's from Sederland. It's class 1. It's white, seedless, and it tells you it's a regal seedless. So it tells you they've actually named this kind of seedless grape. Okay, now watch. Here they turn around and they have to say that it is what the different types of processes that they used. All right, they tell us the weight. They tell us it's produced in South Africa. Okay, and yeah, comes from, it was packed at Tiger Valley, and there is our barcode. But the important process is here that they have to tell us that it has been genetically engineered and what it has been processed from. And then you've got the exact kind of seedless grape. Whereas these guys, they're grapes, and nobody specifies anything. You buy the box, it's got the name of the farm it came from, and it's just one big mega grape. All right, so, and something else, while I'm doing this, Toboko, can you just come up close here? Do you see this? These grapes have got like a sort of a powdery substance on them, okay? People, please make sure that when you buy fruit and vegetables, even if you're going to peel the vegetables from any kind of place where you purchase them from, whether it's a supermarket, grocery store, whatever, make sure that you wash your stuff properly, because very often they spray the crops with all kinds of various wonderful things. And these are the things that can actually affect you and increase your sensitivity to allergies. All right, so always wash your fruit and your vegetables, always, first. And when you've washed it, dried it, or just dried it off lightly, then you can start eating it and doing whatever you have to. And pips from grapes that have seeds, in the hand, in the ashtray, or into a container and in the dustbin, not on your mother's carpet. Take a lesson from me. All right, now, here we have, we're going to have a fruit salad. We have, hey, maybe I'm going to be in a jello, John. Should we start cooking?
Might as well. It's a bit of a cooking show tonight.
Oh. We should do nutrition. We should do breakfast. I used to do breakfast with my boys at King Edward's. They actually make very good breakfasts. All right, people, here we've taken a kiwi fruit. Now remember, a kiwi fruit is a whole cross. This is something that's been interbred, crossed over. They say it's supposed to be a cross between a guava and something. I don't know, this doesn't look like a guava to me. But with all the processes, a brand new fruit has been created. All right, now if you look here, these tiny little seeds there, okay, they can be planted, but the actual fruit itself was something that was engineered. And it was engineered not naturally, but artificially. But they're delicious, what can we say? Then we have this. Now you should all be quite familiar with this. And it's lovely, absolutely juicy. And it didn't work, so let's just peel this off here. Now here I have one big fat pip. And this is a nectarine. And a nectarine is a cross between a peach and a plum, although it's got more peach than plum. But this is a natural occurrence. This is not artificial selection. This is completely selective processing here. And this is over thousands of years. You end up with a fruit that's like this. It has a pip. Inside this pip, you are going to find a seed, and inside the seed, you find an embryo. So if we take this pip and we dry it, and first you eat all the fruit of it, you dry it, you stick it in the ground, and voila, you will have a nectarine tree. Just think of all those pips you've thrown in the dustbin. Just going back to our kiwi fruit here, the fact that they've bred this so that there are little pips, there are seeds, means that it can be planted and it can produce the same structure, and you are going to have a little bit of genetic variation. Remember that all genetically engineered fruits and vegetables aren't all seedless. We want seedless. We want seedless watermelon. There's nothing worse than eating a watermelon and having these bits of pip stick all over the show. Okay? We want it seedless. If it's seedless, it cannot reproduce, people. It can't. So how is reproduction going to take place? Either we are going to use propagation, which means we're going to cut little pieces, or we're going to cut a piece of a vine and stick it on another vine, various forms of vegetative reproduction. Because there are no seeds, you can't have sexual reproduction, so there is no meiosis, okay? There is no genetics, and there is no genetic variation. However, those clever scientists can come up with a kiwi fruit, and they can say, hold on, we're going to breed, mix, breed, this is good, that's good, and they basically speed up natural selection. And you end up with this, and it has its little seeds. But something that's very important is that plants will allow or tolerate polyploidy, whereas animals won't. So if I have 23 chromosomes, 321, number 21 chromosomes in a human, guess what? I end up with a Down syndrome baby. If I take a kiwi fruit and I mess with it and I change it, or a grape, and I change it and I breed it and I do this and I do that and I remove this and I remove that from the chromosomes, guess what? I end up with seedless. See? Absolutely seedless. But I have to propagate. Here, I don't have to propagate. Why? Polyploidy kicks in. Your grapes were bred specifically because they have no pips. We don't want the pips. So they are genetically engineered. The kiwi fruit, genetically engineered, but we don't have a problem with the pips because they're so small. Okay? My favorite, mangoes. Mangoes, they say, are the fruit of the gods. So here we go. First time I've ever cut a mango with a scalpel. Okay.
I don't think it's very ripe either.
And I can see John worrying there about my fingers, and it's not very ripe, so we are sort of going to hack into it a little bit. Not working, so let's try this one. What I want to show you is that there is no hair. Okay. In other words, We don't sort of, I mean, you know how messy mangoes are to start off with. They are incredibly messy fruits to chew or to eat, but oh my gosh, they're so amazing. And it's almost as if the more mess you get on your hands and your face, and you almost need a bib, the nicer it tastes, because it takes us back to being children. But have a look here. There are no hairs, absolutely nothing. I'm going to just give Tebogo this. Okay, if you look here, Tebogo, there's the pip, and there is absolutely no hair here. It's just pure mango fruit. No hairs. So you don't have to worry about flossing after you've eaten this mango. Why? Because those scientists, the guys sitting in their labs or their little white coats that we're all so scared of, they came along and they produced and bred a mango. It's got its seed on the inside, and they've worked on the fact that fruit and vegetables, any plant, can be polyploidy. It can have more than one chromosome pair. So you can add those chromosomes. You can have 6 chromosomes making up a pair or making up a set for characteristics. Okay, so we end up with a delicious, hairless mango. And this mango, you can chew, you can eat, you can make a big mess, but you don't have to floss your teeth afterwards to try and get all those little hairs out. Okay, good for us, yay. But are we messing too much with nature? And I'm going to leave you with that thought, because it's actually a very nice essay question. It makes you think. Remember, they want to stimulate those juices and get your brain working. But that's what I'm going to leave you with. And we, John and I, are going to go for a 1-minute ad break. See you in a minute.
Wasn't that great, guys? I hope you've enjoyed the amazing explanation that we've had on genetic engineering, that you can see it. Go and look in your fruit store. Go and look around you and you'll see there are amazing things happening. Read the newspapers as well. Watch the TV as well. You'll see a whole lot of stuff, arguments and discussion about genetic engineering. Now, I know that genetic engineering isn't the only way things are transformed. So I'm going to hand over to Kathy, and she's got to talk to us a little bit about mutations. So let's get into that, and over to you, Kathy.
All right. People, with regards to artificial selection, artificial selection is what we've just looked at there. You've got your scientists. They literally take the chromosomes, find exactly where the genes are for things that they don't want, characteristics they don't want. They then take genes from other, at least chromosomes and genes from other organisms that have the characteristic they want. They splice, they cut that piece of gene out, or the gene, they splice it into the chromosome where they want it. And that's how it's a huge job. It's very intricate, but that's what they do. All right, so and that's how they make insulin. But we're going to be going into insulin in detail next week. And also, I'm going to put a table for the pros and the cons of genetic engineering. A lot of people feel it is wrong. A lot of people feel if it's going to help people, then it's right. So you've got all your arguments, and we'll put it on because it's a very nice question in an exam. Okay, now, how do we do that? Now, with natural selection is not artificial. It happens naturally over a long period of time, like, for example, our nectarines. Okay, where did they come from? How did they just arrive? Or our peppered moths, the ones with the dark wings and the ones with the light wings. There are thousands of different examples that you can look at, and it's a very nice assignment to give learners to do, and I hope your teachers have, because it tickles your fancy and it makes you interested in a subject when you actually see what's out there and you don't just learn everything in little boxes. So, mutations are very important. And when we did meiosis, the whole meiosis process, and when we did DNA, I explained to you there are 3 types or 3 end results of any kind of mutation. And a mutation is a change in a chromosome, all right, from the gene side. So the gene changes, it changes the chromosome, and then clearly the organism is changed. There are only 3 end results for mutations. One is if it's a fixed mutation. That means it was good. It's helped the organism survive. And the organism is now going to survive. It reproduces and it will pass that gene or that mutation onto the next generation. Okay? That's a fixed mutation. Then we get a lethal mutation. Now, if it's lethal, it kills you. Dead. Okay? Lethal mutation is something happens, it goes wrong, and you're dead. End of story. If you're dead, can you pass those chromosomes and those genes onto the next generation? No. You're dead. The animal's dead. It can't pass anything on because it can't reproduce because, hello, it's finished. That's a lethal mutation. And then we get a neutral mutation. Now, a neutral mutation sits on the fence, literally. And it can't decide whether it's going to flop over this way or flop over that way. Is it going to become fixed or is it going to become lethal? So that mutation sits there, that neutral mutation, and it waits. That's sitting there, and it gets passed on from one generation to the next, until conditions change. And if the conditions are favorable and that mutation assists the organism to survive, it then becomes a fixed mutation, and it is passed on to the next generation. But if the conditions change and that organism, it doesn't help the organism, it actually becomes a lethal mutation, the organism dies and it doesn't get passed on. So your neutral mutation can become fixed and be passed on, or it can become lethal and kill the organism. So lethal, fixed, and neutral. All right, now, what are these? Those are the outcomes, but what are your mutations? You need to know these mutations. And if I was examining you, I would put it in the exam, because I think it's very important for you to know the difference, and it is so easy. So pay attention, and if you're going to get it now, and that's it. You never have to learn it again. You'll never confuse it. You will never have to learn it. It will be stuck in here. And that's what we want, don't we? So here we go. We get my pretty color. Let's go with pink, guys. Sorry. So we're looking at a frame shift mutation. Okay, a mutation is a change in the structure of a gene, which will then result in the structure of the chromosome, or the parts of the chromosome being altered. So look at this. We have a frame shift. Now, what is a frame shift? If you think of the old days, when they had movie cameras, and you had that those big reels. Each of those the reel was made up of little things that looked like this. When they show it at the Oscars, you've got that sort of reel of old movie tape, and they just run it fast. So this would be a picture, and that would be a picture, and that would be a picture of something, and you've got those things down the side here, and it's as it goes through the reel with a light behind it, and then you see these images. All right, those were frames, and that's what they used to be called, frames. All right, so if we do a frame shift and it results in a mutation, it's exactly that. We are shifting the frame. So it's like squashing a picture in here or a picture in here, and it will change the picture we see on the screen. Now, frame shift. How does it work? If we have adenosine, uracil, guanine, and okay, so I'm just going to go the letters instead of saying the words every time. So here we've got AUG, we've got GAG, we've got UCA, we've got ACA. All right, those are our little codons. Okay, there are our codons. Okay, we know that they're the messenger RNA. Why? Because, look, we've got the U's. All right, now that goes and it does its thing, and it's in the whole process. That's what's being read from the DNA. Okay, that's what's being read, coded by the RNA. Now watch here. We go 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. We've got our little codons. Codons are perfect. 3 little triplet bases together, everyone's happy. And something happens, and look at this. This little red guy just comes and he just pushes in. And when he comes and he pushes everyone forward by one shift, one frame shift. Okay? Just squeezes in there. So, now instead of having AUG, I've now got AAU. Ha! Different amino acid. But wait. Now, remember, proteins are made up of lots of amino acids. But wait, look at this. Instead of having GAG, my G from here gets pushed into there. The A from here, or the U from here, and the G from here gets pushed into there. All right? The A gets pushed into there. So each one of these is now going to make a different amino acid. Remember, every triplet base makes one amino, or it gives the recipe for one amino acid. Now, what am I doing? Whoa, I'm changing all the amino acids here in this row, in this sequence, to make a specific protein. So it would be like if I was going to make a cake and I boiled the flour first. Now, can you imagine if you boil the flour first and then you try to make a cake? Not going to work. And that's exactly what you're doing here. The cake being your protein. And what are you doing? You're doing something wrong in that recipe. Frame shift mutation. You have added this little A in, this adenosine, and you've pushed the whole code forward. So now, instead of having that series, you've now got a different one. Why? You've added one. Same applies, they can take one out. So we've got AUG, and come along and they say, wait a minute, A, out you go. Psht. So what happens now? They all move up. And we end up here with only A and U, because G moves there, that G moves there, that A moves there, to try and compensate for this one that's missing. So again, we end up with a whole mess up, and that results in a mutation because you've changed the codons. Right, people, I hope this makes sense. You've moved the frame. You either insert or you delete, but you're only putting in one nitrogenous base, one little base, either A or there, and you're taking it on and off. If we look, I think Belinda said we've got like 1 minute, so I think
it's 5, 4.
Oh, we've got 5, okay. Right, so that's your frame shift. You add one in or you take one out, and all the little triplet bases here that only ever work in threes, they all move across, or they're very polite. Okay, so very polite. So they all just move up one seat. All right, now, your point mutation, okay, is a point. Now, if I make a point, I'm saying this is where my little mark is. That's my point. There it is. Okay? And that's what you have here. It's not a frame shift where you have something that happens at the beginning or the end. You're adding or taking out. Here, a point mutation, something just happens. And during during a DNA replication, something, or transcription, something just goes wrong. And it says, whoa, hold on. And your RNA is now busy transcribing from your DNA. So that's coded from the DNA, okay, onto the RNA. And AUG, GAG, and it says, no, hold on, I'm going to either swap purines of purines, pyrimidines of pyrimidines, or, hello, very daring, swap purines and pyrimidines. And you think, why? What are you doing? And it just says, well, I don't feel like doing this anymore. You know, like you do sometimes when you get homework and you don't feel like doing your homework? That's this. And what do you end up with? A point mutation, which means you mutate your work and you will do badly. We're not going to have that here. So we have frame shift, add one in, and everything moves across. The whole frame shifts. Point, well, instead of guanine, adenine comes in. And wait a minute, instead of adenine, oh my gosh, there's a uracil. And oh, look at this, instead of UCA, the A just gets kicked out and the C takes over. And the same here. Well, okay, look, these are different. You can only need 1 point mutation here to change the entire structure of the organism. But what are they doing? I just showed you different variations. So instead of the A, you have the C. Instead of the A, you have a G. And that's what happens. So in a point mutation, you are changing your purines with another purine. So either, let's do it this way. You're either changing with A to T or to U, all right? And you've got G to C. So they're swapping around and they're just confusing the issue. Or you have it happening that way. And that is your point mutation. So what do we end up with? We end up with a transition or a transversion. And a transversion is, a transition is when a purine swaps with a purine. So the adenine for guanine, or guanine for adenine, or we have uracil for cytosine, or cytosine for uracil. That is a transition, and your transversion is this purine pyrimidine. Hello. So you're going to have 2 different pyrimidines that can come in for a purine, and for a purine you're going to have 2 pyrimidines. So it actually makes it a worse case scenario, and that mutation is bad. Okay, very, very, very bad, and that is what can cause huge issues in a person's life. All right, people, so from simple things to really, really bad things. John.
Kathy, thank you so much. Now, we haven't finished with genetic engineering. So remember, get your questions in, because next week we're going to carry on with genetic engineering. And there's lots more to talk about. This is not an easy subject. It's controversial, but you need to handle it. You need to question it. Post your questions. We'll get back to you. Thank you for being here. Hope you've enjoyed the show and the lesson. You've learned something. Goodbye. Cheers from me and Kathy.